will go to the lumen, and since it is at the lumen of the ER, if it goes through the Golgi and vesicles, it will eventually be secreted to the outside. A precursor oligosaccharide is held in the lumen of the ER membrane by a lipid named dolicol. So the proteins that are in the lumen of the ER are being glycosylated with precursor oligosaccharides. First at the ER, but most importantly at the Golgi, as we will see later. Protein glycosylation takes place first in the rough ER or RER, almost at the time the protein is being translocated. The most complex saccharides, oligosaccharides, are being added at the Golgi. And these are signals for the Golgi to sort them to different compartments. Proteins fold and oligomerize in the lumen of the ER, but misfolded proteins are translocated back to the cytosol. They don't really go through. There are some checkpoints at the ER. So you don't want to secrete protein that is misfolded. So they are translocated back, they are ubiquitinated and further degraded by the proteasome, which is in charge of degrading misfolded proteins. So incompletely folded proteins do not advance to the Golgi. There are lots of checkpoint mechanisms. Lipid biosynthesis takes place in the ER. For example, phosphatidyl choline. And some ERs, depending on the cell type, have specialized functions. For example, in the skeletal muscle, the muscle ER is called sarcoplasmic reticulum. And it's very well known for storing calcium. Calcium is a big player in the skeletal muscle. Or in the liver, the hepatocyte ER is involved in detoxification of hydrophobic toxins. Very specific activities, depending on cell type. So now the Golgi. This is where it is. This is how it looks. It looks like a stack of pancakes with vesicles all around. It's a membrane-bounded vesicles. You can see here. This is the Golgi, very easily recognizable. This is the endoplasmic reticulum here, and this is the nuclear envelope. Okay, it's all associated. Nucleus, the nuclear envelope, the reticulum, which is associated with the envelope, and then the Golgi apparatus, which is connected through the vesicles that go back and forth from the ER to the Golgi. Again, this is the ER, the transitional ER, which is closer to the Golgi. Transport vesicles that go from the ER to the Golgi, and then they are transported back also to maintain the homeostasis of vesicles and membranes. And we have the ergic, the ER-Golgi intermediate complex, the cis phase, the medial phase, the trans phase of the Golgi proper, the trans-Golgi network, and from there, the different vesicles that have different codes or coding. Some of them are coded smoothly and some are not coated or not smoothly coated. And depending on what proteins they have at the surface or what glycoproteins they have, they would go to different places. They would be sorted. So the most complex glycoproteins are being added at the Golgi, and that plays several roles. So first, these are the complex glycoproteins: acetylglucosamine, galactose, and acetylneurominic acid. That is being added through a core region that adds one unit by one and forms a much more complex glycoprotein. The carbohydrate is being added at the lumen of the ER. You will never see a carbohydrate at the cytosol side. It's always at the lumen. The reason for that is that it is meant to protect the protein that is going to be secreted. But you will never see a protein that will be secreted that is at the lumen of the cell. It has to be at the cytosol. It will be seen at the lumen of the ER. All right. The Golgi forms glycoproteins. There is some glycosylation at the ER, but that is very immature glycosylation, very basic carbohydrates. Most of the complex carbohydrates or sequence of carbohydrates are added at the Golgi. And it plays two main roles. One, protection of the secreted proteins, because the protein is less accessible to proteases that are hanging outside of the cell. And two, depending on what's the code, the glycoprotein code, it will go to one place or another. Okay? And this is the sequence of glycosylation. For example, mannose is being added very early at the ER. But then it is removed and is replaced by other complexes, okay? Like uramic acid, galactose, as we have seen. In some cases, mannose is retained and is phosphorylated in a very specific position. And that is a signal for the protein to be taken to the lysosome. So these are like codes, internal codes of a cell. Okay? In some cases, the vesicle fuses with the plasma membrane and releases all the contents. So these are the secretory, this is the secretory vesicle. Is another schematic showing that there are two main mechanisms for the secretion. One is constitutive secretion. It goes, no matter what, it goes, the vesicle goes always to the plasma membrane and releases the contents. Another mechanism is much more regulated. It's regulated by the signals. So the vesicle is just hanging in here, waiting for the signal to tell them to release the cargo. And this is regulated secretion. So we have constitutive secretion and regulated secretion. For example, what happens in mast cells with the hormone or neurotransmitter histamine. Histamine binds, and then that triggers the release of, the hormone binds, and triggers the release of histamine that will produce several cellular phenotypes. So the Golgi. It contains vesicles that are destined to go from the ER to the Golgi. Those ones have a particular code, which is code P2. But the vesicles that go back to the ER from the Golgi, they have to return to keep up with the membrane content. They are code P1 coded. So if you isolate vesicles at one given time, and you look at the code, and these are code P1 coded vesicles, then that will tell you that these vesicles that you isolated were on the way from the Golgi to the ER, right? Because of the code. Vesicles destined to go from the Golgi to the plasma membrane, coming out of the trans-Golgi, they are clathrin coded. Okay? These are codes that will tell you where these vesicles are going to. In the Golgi, formation, processing, and maturation of the glycoproteins take place. The addition of oligosaccharides to the protein makes it more resistant to proteases. There are plenty of proteases outside of the cell, so they have to be protected. The vesicles are not just going randomly within the cytosol. They are transported by what is called motor proteins, like dynein, kinesin, that walk along the microtubules. Microtubules made out of tubulin. This has been studied quite well, and phosphorylation of the mannose residue targets a protein, for example, a hydrolase, to the lysosome. So this is the signal to tell a particular protein to go to the lysosome. In the lysosome, the pH is acidic and the hydrolase becomes active. So hydrolases are meant to work only in the lysosome, right? But they have to be transported through different stages. So nature is so wise that made them inactive when the hydrolases are in neutral pH, for example. But when they go to the lysosome and there is a different pH, more acidic, then the hydrolase becomes active and starts disaggregating some proteins within the lysosome. So in vesicular trafficking, there is an orderly process, which is budding, docking, and fusion. And all these processes have been very well studied. They're very, very complex. There is not just one or two or three, many proteins that are involved in these highly regulated processes. And finally, secretion to the exterior of a cell can be constitutive or regulated. As I mentioned, example, histamine in mast cells. Okay, so now, cellular eating and drinking. Endocytosis, drinking, eating, phagocytosis. endocytosis could be through two different ways. Fluid phase endocytosis or pinocytosis, or receptor-mediated endocytosis. There has to be a receptor at the surface of what is called a pit here. Phagocytosis is a complete different process that involves vesicles that are much larger than the endocytic vesicles, like 10 times, 5 to 10 times. So endocytosis can be constitutive or receptor-mediated, like the very famous example of the LDL for cholesterol homeostasis. Phagocytosis is performed by professional phagocytes like macrophages, dendritic cells, neutrophils, and involves the eating of entire organism, entire microorganisms like bacteria, etc. There has to be, the vesicle that is formed has to be much bigger than the endocytotic vesicle. This is phagocytosis. There's also one process which is called autophagy, or self-eating. So the phagolysosomes, or the phagocytic vesicles, are not only for degradation of proteins that come from outside, but also from proteins that come from inside, or structures or organelles that come from inside. For example, an entire mitochondria can be degraded by autophagy. For example, when the mitochondria is old enough and is prone to die, the best way to get rid of the mitochondria is through self-eating or autophagy. Okay? So the whole endocytotic process is very orderly organized. There's an early, late endosome and lysosome finally. And through this process, the pH becomes more acidic. So it starts slightly acidic and it keeps going into more acidic pH. The lysosome products can be utilized by the cell or simply secreted. A process that is called desiccation. Some endosomes, though, undergo transcytosis, which is traffic to the opposite side of the cell. Vesicles are fused to the plasma membrane, and their contents released to the exterior. This is called transcytosis. So this is a depiction of, for example, phagocytosis, when a bacterium, an entire bacterium, is devoured by the cell, okay? By professional phagocytic cells, like macrophages. Through phagocytosis, it goes into phagosomes, and eventually everything ends up at the lysosome. Could be endocytosis, here, with an early endosome, late endosome, and finally a lysosome, or internal phagocytosis, or autophagy, okay, in which an old and prone to death organelle, like the mitochondrion, because everything has some lifetime, okay, some half-lifetime. When a mitochondrion has been used a lot, then it has to be replaced by another one. So the best way is to get rid of it by self-eating, autophagy, out of phagosome, which will end up again into the lysosome. So this is how endocytosis begins in coated pits. This is the plasma membrane that makes this bend here. It forms the pit, and the pit closes and forms the vesicle. Looks simple, but it's very complex, and it involves lots of different products. In fact, clathrin, that is clathrin complex, that is this structure of proteins here outside of the pit, bind the pit here. They have a structure that sort of allows the pit to bend and forces the pit to bend even more. Finally, forming the vesicle, the entire vesicle that pinches off from here. Okay? So there's a lot of fusion events involved and separation of membranes, very tightly regulated. So this is an example, an experiment has been done regarding cholesterol processing or metabolism, in which a receptor of the LDL, which normally binds to the clathrin here, the clathrin-coated pit here, has a domain that binds to the pit. They eliminated that sequence, and the pit forms, the vesicle forms, but it's devoid of the cargo, devoid of the LDL. That means that this unit here, or this region, the protein that binds to the clathrin, is necessary for the cargo to be loaded